Okay, so I will tell you about pre-mating, so female receptivity circuits, and I'm interested to understand how the brain incorporates information from sensory stimuli to understand the environment. So this is a project I've done in collaboration with Istvan Thijs, a talented PhD student in our lab, in Greg Jeffries' lab. And the title is Generating Parallel Representations of Position and Identity in the Olfactory System. You see here in this lovely illustration by our visual aid department, a female fly who is localizing a male fly using those bubbles which represent automolecules. So we found that females use the smell of a male to not only recognize the male, but also to localize the male. And we're looking into the mechanisms of how this is done. Okay, so what you see here is a visual scene when I took my child to go climbing in a climbing gym some weeks ago. And it brings me to the question of how does the brain extract information from a sensory stimulus? So let's think about this image as a visual scene and try to think about what the brain actually does to extract information from a sensory input, this visual scene. So when I'm looking on this image, there are a lot of different types of information that I can gain from this. So there are the colors, the shapes, the size of objects, the location and movements, a lot of different axes that the brain can encode information. And each of those components or axes can be encoded separately and eventually combined with other sensory information, maybe from other senses, to reach an understanding of a concept or an entity. So for example, the shapes and the colors could be united with some information from auditory or olfactory or other senses that would give me the perception that this is my son, and other information such as the location and the movement, so more spatial information, could tell me that something is descending. That happened when my son decided to drop from the wall and go down. And by combining this information, eventually after the brain had encoded individual pieces of information, we can reach an understanding of what to do. So this is my son and he's falling, and then I would know to open my arms and do something about it. So I'm interested to know first about the initial steps. So how does the brain take a complex sensory scene and breaks it down into components? So we're looking on pheromone perception. The first studied pheromone is the silk moth pheromone. So here you see an image of a female silk moth, and the male is around her. So the male is very attracted to the female pheromone, and he's using this pheromone for two separate tasks. So first is to recognize and identify that this is a female moth from the right age and the right species, but also he's using the pheromone to track the female location, so those males can fly miles away from very few molecules of odor. And I wanted to then look on a similar question using Drosophila to see how the brain can extract this information from Drosophila pheromone. So in Drosophila, the famous pheromone is CVA. It's not a female pheromone like the moth, but it's a pheromone produced by males. And we already know that it has sexually dimorphic effects on behavior. So it is used for object recognition. In that aspect, the male pheromone is attractive to female and makes them want to copulate. And the same odor is aversive to males and make them want to fight. So we already know that this pheromone is used for sexually dimorphic behaviors, but we don't know whether this pheromone is also used to track the location of other males. So that was the first thing I wanted to look into. Can CVA serve to track male location? How does the brain extract both the positional and qualitative features from a single odor? And how is this information processed in the brain to instruct behavior? So this is the head of the fly, and on the antenna are the receptor neurons, the RNs, which are the first relay station that get information about the odor. And here we put the female under a microscope, and we have a male on a stick, basically a micro manipulator that we can put closer and farther away from the female and image the neuronal responses of the receptor neurons. And as you can see here, the distance from the female and the neuronal activity. So of course, the closer the male is to the female, the activity is stronger, but it also showed us that the CVA operates in a very narrow range. So 5 millimeters away from the female, there is already no activity in the receptor neuron. So it's like the male is walking around with a halo, which is only 2 fly sizes in range. So it's a very narrow range, which is a little bit surprising if you think about the strong effect that this pheromone has on behavior. So after we saw this, we wanted to know whether females really care about being within this sensation range, within 5 millimeters away from the male. So I put a decapitated male in this arena, glued to the floor, and one live female walking around it. And I measure how long the female spent time inside this 5 millimeter range around the male, so the CVA sensation range, compared to the time that she spent outside this range. And as you can see here, females prefer to be close to the male. So this is done in complete darkness and the male is dead. So the only sense that they're using here is the olfactory sense. Female prefers to be in proximity to the male, whereas males avoid this range. And the preference depends on intact olfaction, because using orchomutants that cannot smell completely impair this preference. So female want to be within this sensory range. Now we wanted to look more closely into the activity of the receptor neurons. So in this setup, we put the female under the microscope, but instead of a fly on a stick, we have a live walking fly around the female. So this is the male. And we measure the activity of the receptor neurons in this top line here, and also the distance between the male and the female. As you can see, the neurons are activated when the male is close to the female. And analyzing this data, we see that there is a preference of the neurons to respond when the male is in front of the female. And also we see a side preference. So looking on the difference between the left and right antenna, we see that the left antenna is mostly activated when the male is on the left side. The right antenna is mostly activated when the male is on the right side. So there is both a front bias and a side bias. So already we can see that the information from the receptors can be used to localize the spatial information and location of the male. So there is an odor gradient between the left and right antenna. Can flies use this odor gradient to orient in space? To answer this question, I put a male and a female freely walking in this arena, and I measure if the female made turns when she entered this sphere where she can sense the CVA smell. So I looked on the orientation of the female within CVA sensation range. And as you can see here, the location of the male when the female initiated a turn is quite uniformly distributed between left and right. But when we remove one antenna, we start to see a bias towards the side of the intact antenna, which was significant. So the females do use the gradient between the left and right to orient in space. And if you remove one antenna, they get this side bias. And this was true both for males and females. So also for males, ablating one antenna led to a side bias. So females use the odor gradient to navigate, and we wanted to know when they do make a turn, is the turn towards the male or away from the male? In order to know that, we put the fly on a ball, and we presented again the male on the micromanipulator, either on the right side or the left side, and measured the female movement when the male was presented. And as you see here, the female made mostly movements towards the right when the male was presented on the right, and movements towards the left when the male was presented on the left. So female turned towards the male. And this was true in complete darkness. When we turn on the light, the females avoided the male. So they have different like alertness response that they avoid when something approaches them in the light, but they are attracted to the CVA smell when it's happening in complete darkness. So to this mid-summary, I hope I could convince you that pheromones are used not only to recognize the object, but also to track the location. I showed that CVA is a short-range pheromone, only with 5 millimeters around the female. Females prefer to be within the CVA detection range, and this requires intact olfactory receptors. We saw lateral bias in the neuronal activity that reflects a gradient between the left and right antenna. Females use this gradient to orient towards a male, and when they do turn, they turn towards male in response to a presentation of the male on one side. Okay, so now we want to go into the nervous system and try to see how the fly brain represents all of this information that comes from the smell, both the recognition of the object and tracking the location. In order to do that, I'm using connectomics. So luckily, Megan already introduced you to what connectomics is. So we have a brain of a single fly, and we can really follow and reconstruct all the individual neurons from this electron microscopy volume. So you see in this nice video, all the different neurons, and initially manually we reconstructed neurons one by one, but eventually using machine learning algorithms, we can now reconstruct full connectomics data sets. This work was done in collaboration with Genelia and a lot of contribution from our lab, Drosophila Connectomics in Cambridge, and Greg's lab in general. So we have this magnificent data set of many, many neurons in the fly brain. And so I started looking into the receptor neurons to see what is downstream. So we found those two pathways. I call them 2A and 2B. So these are the two types of projection neurons downstream of the receptor neurons. And 2A neurons are very canonical, famous projection neurons, but those 2B neurons are novel. So they were not described before we started this project. So first we wanted to see that these neurons actually respond to CVA. So indeed they do. And interestingly, these two pathways project to different brain regions. So that made us think that maybe they have different roles in encoding information from the smell. And I wanted to know what are the roles of those parallel pathways, both in sexual behavior and in male localization. In order to look on sexual behavior, I put a pair of freely walking flies, males and females, and I track their activity and classify their sexual behavior. So here, activating 2A or 2B neurons, I'm looking on a copulation proportion as a measure for female receptivity. So looking how many pairs of flies mated with time. And as you see, activating 2A neurons with optogenetics didn't increase female receptivity, but activating 2B neurons did increase the proportion of copulating pairs, meaning females were more willing to mate. So 2B neurons seem to be important for female receptivity, and also ablating 2A neurons had no effect on female receptivity, so they are the same like control, but ablating 2B neurons did impair female receptivity. So those two parallel pathways have distinct effects on sexual behaviors. In females, only 2B neurons control sexual behaviors. Now, looking on the other effect, which is known, a behavior effect, which is known for CVA, is male-male aggression. And I saw when I activated 2B neurons in males, it increased male aggression, but activating 2A neurons did not. So both in females and in males, the 2B neurons seem to be the ones critical for those phenotypes that were described in the literature, male-male aggression and increased female receptivity. So if those 2B neurons are responsible for the object recognition, what do 2A neurons do? Maybe they are responsible for tracking the location of the male. In order to do that, we went back to our paradigm where we image from the female with a freely walking male around her, but now we're imaging from 2A neurons. And as you see here again, the top line is the response of 2A neurons to the neuron activity, and the bottom line is the distance between the male and the female. And again, we see that proximity of the male drive neuronal activity, but we can also see that the left neurons are mostly active when the male is on the left, and right neurons are only active or mostly active when the male is on the right. So we see an increase in the side biases. And this was a bit unexpected. So if we're looking on the data collectively, Here, the first layer is, the first line is from the receptor neuron, and the second line is the results from 2A neurons. And we see an increased front bias, but also an increased lateralization. So the 2A neurons on the left side respond mostly when the male is on the left, and vice versa. And this is stronger than the lateralization in the receptor neurons. In order to really understand that phenomenon more, we went back to the male on a stick paradigm, where we image always from the same side of the brain and present the male either from the ipsilateral side of the imaging or from the contralateral side. And these are the responses in the receptor neurons that are stronger in the ipsilateral side of presentation than the contralateral side. And this reduction is our measure of the lateralization, so the side bias. And measuring from the 2A neurons, we see that the lateralization is increased, as quantified here. And as I started saying, this is a little bit surprising, because if you look into the connectome and what we know about the nervous system, the projection neurons receive information from receptor neurons in both sides, the ipsi and the contra. So if anything, we expected that they would have a lower lateralization degree than the receptor neurons. So the receptor neurons are sitting in the antenna, and due to the distance between the left and right antenna, you could expect some gradient, even though the antenna are only 250 microliter different. So the distance between them is very short. But even so, it is enough to create a gradient. But you would expect that the flow of information onto the projection neuron would diminish this gradient. But instead, we see that it's getting bigger. So we thought that there must be some active component that blocks the information from one side to increase the contrast between the left and right. So we went into the connectome and looked for such a component, and we found those local neurons that specifically receive information from the contralateral receptor neurons and then block the 2A neurons. And we found a line for them and imaged this line, and indeed those neurons mostly respond to the presentation of the male from the contralateral side as expected from the connectome. And when we block the contralateral antenna, we see that the lateralization is reduced. So we really need the information coming from the contralateral sides to actively increase the contrast between the sides. So side bias inhibition by a local neuron increased the bilateral contrast in 2A. Looking further into that, we blocked those local neurons using a chloride channel, so a histamine-gated chloride channel, and we saw that blocking those neurons reduced the lateralization in a temporary manner. So after you wash away the histamine, the neurons go back to normal. So another proof that those neurons actively block information from the contralateral side. Now, those neurons are very interesting because, as you see, they innervate the entire antennal lobe. So they're not a specific local neuron, but rather a general one. Yet the effect that they have on blocking contralateral information is quite local, and it doesn't spread to any other compartment. So we think that they operate in a compartmentalized way, such that each of the glomeruli is serving as its own mini circuit that can be, the gain can be controlled either to increase or decrease the information coming from both sides. And looking into the connectome, we see that there is a strong degree of lateralization in the way that this neuron connects to many different glomeruli. So if you look here, the ratio of input from contra and ipsi. So for example, the DA1 glomerulus, which receives the CVA information, has 5 times more input from the contralateral side than the ipsilateral side onto this LN. So that's this axis. But there are even neurons, glomeruli, that receive stronger lateralization. And here is the percentage of input onto second-degree neuron, onto PN. So a lot of glomeruli get lateralized information through this LN, which could increase bilateral contrast across multiple glomeruli. Next, we wanted to see, can the CVA gradient across left and right be used to encode the position of the male? So we presented the male in 18 different locations around the female, an image from 2A neurons, either on, in both the left and right sides. As you see here, the cyan is in the right hemisphere and the red is in the left hemisphere across all of these positions. And we saw, of course, that, again, the responses in the right hemisphere were stronger when the male was on the right and vice versa. But also, we saw that the responses are very directly related to the location or the angular position of the male. So we were able to build a model that used both the sum and the difference of the PN activity to predict male angular position, as you can see here. So the information from this neuron is enough to very precisely encode the location of the male in space. So going back to the connectivity diagram, 2B neurons are responsible for object recognition, whereas the information in 2A is enough to encode the location of male in space. And imaging those neurons, we see that the activity in 2A neurons has an on response, so it's phasic, whereas the 2B neurons has a sustained response. And this is quite not atypical for PNs to be responsive for 10 seconds. So here we put the male next to the female for 10 seconds, and the response of the neurons stay on all the time. So they really represent the presence of a male, like more like an analog readout of the amount of CVA, whereas 2A neurons are in a position to encode changes in changes in concentration of the neurons, changes in concentration of CVA rather than absolute amount. So they are more suitable to encode movement, location, and changes. Now we want to go downstream into the third layer to see where the information is going from there. And from this nice scheme with just 2 neurons downstream, we now funnel out to multiple neurons downstream of the second order neurons. So 2A neurons have 40 partners and 2B neurons have 11 partners. So there is a great diversity where this information is going into. And so we wanted to take a few examples and try to really follow up and see how the brain extracts information both about the position and the identity of this odor, and really how it's been encoded. And here is a nice video where we can see, I haven't shown really the anatomy of these neurons, but here you see the receptor neurons, and now you see the 2 different populations of projection neurons, 2A and 2B. And now in gray, you see all the different downstream partners in the third layer. So it's really like a forest of many, many different types of neurons. So this was quite confusing for us when we first saw this, because it was nice to have this labeled line pathway, but obviously the brain doesn't work like that. So even information that starts from a very precise entry point, spreads out to half the brain sometimes. So so it was an interesting question to see really what are all these neurons doing. So I'm going to tell you about 3 neurons that we really followed up to see what is their role in encoding the information. Okay, so 3A and 3B neurons. 3A are downstream to 2A neurons. They're called AV2A2, and 3B are downstream to 2B neurons. They are the PC1 neurons that Megan already introduced to you, which we know play a role in female receptivity and encoding the mating status. Okay, so looking onto those 2 pathways separately, the 3A neurons are sexually isomorphic neurons, so they are the same between males and females, whereas 3B or PC1 neurons are sexually dimorphic neurons. 3A neurons have phasic responses to the smell of a male. So similarly to their input neurons, they only respond at the beginning of the odor. This is even stronger than the response of 2A. So really, when the male is very close to the female, we don't get any response. So they only have a phasic on response, and then they're quiet. Whereas the PC1 neurons stay on, so they have a sustained response to the smell of a male. 3A neurons had no effect on female receptivity, whereas activating PC1 or 3B neurons did affect female receptivity, as was already shown in the literature. But interestingly, the speed of the male movement is encoded by those 3A neurons. So when we presented the male in increasing speeds towards the female, the activity in these neurons was stronger. So they have a speed-dependent response. So we want to correlate this or to relate that to the what and where streams of information in the mammalian visual system. So we see here that similarly, we have a pathway that represents motion and spatial relations, and we have another pathway that separately represents identity, form, color, recognition. And those parallel pathway work to encode specific information from the same stimulus. Another downstream partner is 3C neurons, also called ASPG, which are studied in our lab for some years now already. So those are also sexually dimorphic neurons. And I wanted to know if they have any effect on femoral receptivity. So we found a line that specifically labeled those neurons and activated them using optogenetics. And we found that activating those neurons strongly increased femoral receptivity. Now, this was a bit puzzling because the input neuron 2A had no effect on femoral receptivity, if you remember from my earlier slides. So how come the downstream partners do have an effect on femoral receptivity? We thought maybe they receive information from another source. So we went back into the connectome, and indeed we found that 3C neurons receive information also from neurons that come from the proboscis that bring information about the taste. And we wanted to know what exactly they're testing. And so we presented an array of different taste tests to the proboscis of the female. And we found the strongest response was to male genitals. So this pathway integrates across the smell of a male and the taste of a male to lead to a sensory integration and perception of a multisensory scene or context. And if I tried to activate either of those pathways individually, I didn't see any effect on femoral receptivity. But if I activate both the taste and the smell pathway at the same time, this red line here, then I do see an increase in femoral receptivity, which is similar to activating their downstream partners. So really, this tells us that those neurons serve as a coincidence detectors, which you could also see from the response pattern. They only are operating in a very short time window. So you need not only that the smell will be around in the air, but the flies need to smell the male and taste the male at the same time for this pathway to be activated and trigger a behavioral switch into a state of receptivity. Now, we don't really know when this happens because usually in female, during the courtship, the females don't really taste the males. It's not something that they do. The males do taste the female, they tap them and they lick them, but during courtship, the females never turn around and taste the males. So I'm thinking that this probably happens when the females taste the samples of pheromones that males leave around them. So during courtship, males produce pheromone droplets, which could provide both CVA and taste molecules that would lead to activation of this pathway and to increase femoral receptivity. So this is an ongoing project that we want to start in the lab. Okay, so interestingly, we have these two pathways that both contribute to femoral receptivity. And I wanted to know whether they are operating in parallel or do they depend on each other. So for example, if both 3C neurons and 3B neurons are required independently, then activating either of them would increase femoral receptivity. But if downstream there is some serial structure, then if I activate 3C neurons while 3B neurons are ablated, I wouldn't be able to see an increase in receptivity. So in order to do that, I devised an epistasis experiment. Here you see the purple line is activation of 3C neurons or ASPG neurons, which again increase femoral receptivity. And here the yellow line is ablating PC1 neurons, which reduce femoral receptivity, but the black line is activating 3C neurons on top of ablating 3B neurons. And we were able to restore part of the receptivity, not all of it, but part of it we were able to rescue. So this tells us that at least to some degree, those two pathways are running in parallel or independent to each other. Interestingly, both of those neurons also participate in female-female aggression. So that was one of the first things that I found when I started this project, that activating 3C neurons increased female aggression, as you see here. Now, normally, virgin females don't tend to be very aggressive. before they are mated. So this was a very surprising finding. And while I was doing this project, some really cool results came from Katie Streiter in Jerry Rubin's lab that showing that PC1 neurons are also leading to female aggression. So it's interesting to see that those parallel pathways, both of them activate both female courtship or mating response and female-female aggression. And if you're thinking about it, there is this model from David Anderson's paper in 2016, which I'm sure some of the organizers are very familiar with. We can think about the control of courtship and aggression either as being controlled by separate streams of separate neural circuits, or we could think about aggression and courtship or mating responses being controlled by a joint hub, an integration of sensory cues and internal state that could lead to either increased aggression or increased mating. But I think it's a little bit more complex than that, because even within PC1 neurons, we know that there are different subtypes that control different aspects of the behavior. So even though I do see that activating ASPG neurons or 3C neurons do increase both aggression and female receptivity, there still could be difference with individual subtypes of neurons that could promote distinct behavior. So, but I think this is a very interesting question to look into. So to conclude, we found that CVA plays a double role, contributing both to immediate spatial orientation, that is the where, and also to long-term sexually dimorphic behaviors, that is the what. We found that CVA is used as a positional cue via an active mechanism of side bias inhibition in the antenna that enhances bilateral contrast, not in antenna, sorry, in the antenna lobe. So that actively increase the contrast between the information coming from both sides, so to make the flies more sensitive to very narrow gradients, even in a very narrow range. And in the third layer, we found multiple examples of feature extraction. We found a speed sensor for the approach of a male. We found a tonic response to the presence of a male. And we found a multisensory coincidence detection of a chemosensory scene that incorporates information both from taste and smell to increase female receptivity. So going back to the original slide that I showed you, if we're thinking about how the brain extracts information from a specific sensory stimulus, and looking on the data that I shared with you today, we see that the brain can break the information from the smell of a male fly, and some of those features that represent the location and movement are going into 3A or some specific pathways, while other information, maybe about the shape and about the identity of the male, is going to other a neuronal pathway, and some of those pathways also integrate information from other senses, such as 3C neurons that bring also information about taste. And eventually, knowing that this is a fit male and that this male is approaching should lead the female to make a decision as to the appropriate behavior, which would be either to accept or to reject this male and to agree to copulate. So I would like to thank Greg Jeffries and everybody in the Jeffries lab, first and foremost Istvan, who did a lot of work for this project, Daniel Munch in Carlos Ribeiro's lab at the Champalimaud for doing the taste presentation experiments, those great people from Connectomics to do a lot of manual reconstructions of neurons before we started using automatic reconstructions, but also doing some great analysis on the data. Eric Kupfer and Keta Sahina, former Anderson lab members, for helping me to set up the behavioral arena when I first started in the lab, and those people from fly community for providing fly stocks, and of course the funding agencies. Thank you.